Have you ever stopped to wonder if the tough times faced by your family in the past, or, even big changes in society, could have somehow left a mark on you? Not in your genes themselves, but maybe in how those genes behave.
It's a really mind-bending idea, isn't it?
It really is, and it's exactly what we're diving into today,
Yeah.
something called epigenetic drift.
That's right. We're going to explore how our culture, our environment, how it can become, well, surprisingly intertwined with our biology through this fascinating process.
Yeah, and the information we're looking at today, it comes from a stack of scientific reports, research papers, all focused on this world of epigenetics.
hmm
Now, if you're new to this, maybe think of epigenetics as like a second set of instructions for your DNA. It doesn't change the basic code, right?
No, exactly.
But it tells those genes when and how to be active. It's like having the same musical notes, but different players interpreting them, sort of.
That's a great analogy. And what's truly amazing, really, is just how sensitive this epigenome, the second layer, is to what's happening around us, and even potentially to what happened to our ancestors.
Wow. Okay, so our mission today is basically to cut through this, the scientific jargon
Hmm.
and pull out the most important insights about this epigenetic drift, how it connects to culture, environment, all of that. We want to give you a clear path to understanding this really vital area of biology.
Sounds good.
Okay, so let's get started. Let's look at the basics, the fundamental principles of epigenetics first.
Okay. So, in simple terms, epigenetics is about changes in how our genes work.
hmm
Changes that can be passed down when cells divide, and sometimes maybe even across generations.
Right.
Crucially, though, without any changes to the actual DNA sequence itself.
Hmm.
As you said, it's like a second layer of information, the epigenome,
Yeah.
telling the DNA how to be read and used.
And this isn't some like out there concept, right? Epigenetics is absolutely essential for just
Mhm.
normal development.
Oh, complete.
I mean, every single cell in your body has the same DNA, but epigenetics is the reason why your skin cells are so different from your brain cells, for instance.
Precisely. It's these epigenetic mechanisms that carefully control which genes are active and which are silent, in different cell types at different times.
Mhm.
It orchestrates the whole thing.
Okay.
But what's fascinating here and really key for our discussion is that this epigenome isn't fixed. It's incredibly responsive. It changes based on all sorts of environmental cues.
Like what kind of things?
Well, think about the food we eat, any toxins we might be exposed to, even how much stress we're under. All these things can leave their mark.
Okay, so how does this actually happen on a tiny molecular level? What are the key players, the mechanisms involved?
Yeah, so there are three main ways this happens that we really need to get our heads around. First, there are these direct chemical tags on the DNA itself called DNA methylation.
Okay.
Second, there are modifications to the proteins that DNA wraps around. These are called histone modifications.
Right. Histones.
And third, there's the action of these small regulatory molecules called non-coding RNAs. And often they all work together, interconnected.
Like an orchestra, you said earlier.
Exactly. So let's start with DNA methylation. This basically involves adding a tiny chemical group, a methyl group, onto one of the DNA building blocks, usually at specific spots.
And enzymes do this.
Yep. There are enzymes that add them and others that can remove them. And generally speaking, when these methyl tags are added to the control region of a gene, what we call the promoter,
Mm-hmm.
it often makes that gene less active. It sort of silences it, like turning down the volume. Though sometimes methylation in the main body of the gene can actually boost its activity. It's complex.
Okay, so methylation is like putting a dimmer switch on a gene, usually in its control region. Got it.
Yeah.
What about these histone modifications? That sounds a bit more involved.
It is a bit more intricate, yeah. But think about how your DNA is packaged inside your cells. It's not just a loose string, right?
No, it's wound up.
Exactly. It's wound tightly around these proteins called histones. This whole structure is called chromatin. Now, these histones have little tails that stick out,
Okay.
and these tails can be modified in lots of different ways. Chemical tags get added, like acetyl groups or methyl groups.
Yep. And that changes things.
Big time. These modifications affect how tightly the DNA is wound around the histones.
Mm-hmm.
Adding acetyl tags, for example, usually loosens the winding, makes the genes easier to access, turns them on.
Oh.
Removing those acetyl tags tightens things up again, which can silence the genes. Methyl tags on histones are trickier. They can either activate or repress genes depending on exactly where they land and what other tags are nearby. It's like a code, the histone code.
So DNA methylation is maybe more like a direct volume control, while histone modifications are more about how easy it is to even get to the gene's on switch in the first place?
That's a pretty good way to think about it, yeah.
Yeah.
And then number 3, we have the non-coding RNAs, or ncRNAs.
Right, RNAs that don't make proteins.
Exactly. They aren't used as blueprints for proteins, but they have really crucial regulatory jobs. Take microRNAs or miRNAs, they're tiny, and they often silence genes after the gene's message has been made.
Ah.
They can either cause that message to break down or just block it from being used to make a protein. Then you have long non-coding RNAs, LNCRNAs, much bigger, much more diverse group.
What do they do?
All sorts of things. They can act like scaffolds, bringing proteins together to regulate genes. They can guide those histone-modifying enzymes we just talked about to specific spots on the DNA. They can even act like sponges, soaking up those microRNAs.
Wow.
And there are others too, like piRNAs, mainly in reproductive cells, helping keep the genome stable. But the key thing is how interconnected all this is. ncRNAs influence methylation in histones, and methylation in histones influence ncRNAs. It's a whole network.
It sounds incredibly complex, but also really dynamic. So how stable are these epigenetic marks? Can they be changed? And crucially, can they actually be passed down?
That's the million-dollar question, really. And the answer is, it's complicated. While these epigenetic patterns are set up and maintained throughout our lives, they're also designed to be flexible. They can change in response to development environment.
Okay.
We have cellular machinery that copies these patterns when cells divide, which is vital for maintaining cell identity, like making sure a liver cell stays a liver cell. But importantly, many marks are reversible.
There are eraser enzymes.
Exactly. Enzymes that erase and rewrite marks. That's a huge area for potential therapies, trying to reset faulty epigenetic patterns.
Right. But what about pass them down across generations? Parent to child, meiotic inheritance.
Yeah, that's where it gets particularly complex, especially in mammals like us. There's a major reset process that happens in our reproductive cells, sperm and eggs, and again, in the very early embryo.
A wipe clean.
It was thought to wipe the slate clean of most epigenetic marks, yeah. But the evidence is mounting now that some regions, or maybe some specific types of marks, can actually escape this reset. Or they get reestablished in a way that reflects the experiences of previous generations.
So the idea that what our grandparents went through, say, extreme stress or famine, could somehow influence our biology today through epigenetics, it's not completely out of left field then.
The research is definitely pointing in that direction, yeah. Though the exact mechanisms are still being figured out. And one really key insight from our sources is the critical importance of the first 1,000 days of life.
From conception to age 2, roughly.
Exactly. That period is a window where the epigenome seems particularly open, particularly sensitive to environmental influences. What happens then, nutrition, stress, exposure, can have really long-lasting impacts on health.
And life isn't simple, right? Environmental challenges often come together.
Absolutely. It's likely that things like toxin exposure plus poor nutrition plus stress trigger a whole cascade of changes across these interconnected epigenetic layers, methylation, histones, and ncRNAs, potentially leading to more stable multi-layered spars, or what we could think of as epigenetic memories.
Okay, so we've got a handle on the basics. Now let's dive into one of the most, compelling and maybe controversial areas. This idea that trauma experienced by previous generations could somehow affect us through these epigenetic mechanisms.
Yeah, this is a truly fascinating area, and as you say, actively debated. Well, but our sources do present some pretty compelling evidence from studies in both people and animal models, suggesting that significant life experiences, especially trauma, might indeed have biological effects that ripple across generations, possibly via epigenetics.
Let's start with the human studies. The work of Dr. Rachel Yehuda on Holocaust survivors and their children feels really central here. What did her research actually find?
Right, Dr. Yehuda's team looked specifically at whether the effects of the extreme trauma experienced by Holocaust survivors could be detected biologically in their adult children decades later. They looked at psychology, hormones, and epigenetics. A key finding involved a gene called FKBP5. It's crucial for regulating our body's stress response system, the HPA axis. They found altered patterns of DNA and methylation in this gene, in both the survivors and their offspring.
The same changes.
Interestingly, no, not always. That's what's really intriguing. In some specific spots on the gene, survivors tended to have more methylation compared to controls, while their children had less methylation in the very same area compared to control offspring.
Wow, opposite directions. What does that suggest?
It suggests a really complex intergenerational response. Maybe not simple inheritance of the same mark, but perhaps some kind of biological adaptation or reaction in the next generation to the parent's trauma experience or maybe the environment the parent created.
That is really fascinating. So the children's epigenome is almost reacting specifically. What about other genes related to stress? Did they look at those too?
They did. They also looked at the NR3C1 gene. This gene makes the glucocorticoid receptor basically the docking station for the stress hormone cortisol.
Okay.
And they found that the level of methylation in a key control region of this gene in the offspring was linked to whether their parents had PTSD. And these methylation patterns, in turn, were correlated with how the offspring's bodies regulated cortisol.
And this connects to health outcomes.
Well, it's known that the children of Holocaust survivors, as a group, do show a higher risk for conditions like PTSD, depression, anxiety. And they often have distinct patterns of stress hormone regulation. This epigenetic link provides a potential biological mechanism contributing to that vulnerability.
So it's how the genes are regulated epigenetically, tied to the parent's trauma, and potentially the children's vulnerabilities. The Dutch hunger winter study is another really powerful, almost tragic example, isn't it? How a huge environmental event can have these lasting, even multi-generational effects.
Absolutely. The severe famine in the Netherlands near the end of World War II, it was devastating. But from a scientific viewpoint, it acted as a kind of natural experiment. Researchers have followed individuals who were developing in the womb during that famine for decades.
And what did they find epigenetically?
One really significant finding focused on a gene called IGF-2, which is vital for growth. Individuals who were exposed to the famine very early in pregnancy, like around conception, showed persistently lower levels of DNA methylation at a specific control region of this IGF-2 gene. And this difference was still measurable 60 years later compared to their unexposed siblings.
60 years. And the timing mattered.
Critically, this effect was specific to that very early exposure window. Exposure later in pregnancy didn't seem to have the same lasting impact on this particular gene's methylation.
And did this epigenetic change translate into actual health consequences for these people later on?
It seems so. That early exposure group had increased risks later in life for things like metabolic conditions, obesity, diabetes, cardiovascular disease, and also for certain mental health conditions like schizophrenia and mood disorders.
And I think the sources mention possible effects even further down the line.
Yeah, there's some suggestive evidence, though maybe less definitive, hinting at transgenerational implications. Things like altered birth weights or adult weights in the grandchildren, the F2, and maybe even F3 generations, depending on whether it was a mother or father who was exposed in utero back during the famine.
Wow. Okay, so beyond these really specific major events like the Holocaust or the Hunger Winter, our sources also mention other human studies linking, say, prenatal stress or difficult childhoods to epigenetic changes, right?
That's correct. There's definitely a growing body of research. Studies have connected maternal stress or depression during pregnancy to epigenetic changes in the baby, often in genes involved in the stress response system, like that NR3C1 gene again.
And childhood adversity.
Similarly, yes. Studies looking at individuals who experienced abuse or neglect early in life have found epigenetic alterations, for instance, in brain tissue related to stress regulation, sometimes even linked to outcomes like suicide. And even in combat veterans who develop PTSD, researchers are finding epigenetic changes in stress-related genes in their blood cells.
It's quite sobering, isn't it, to think how these profound life experiences can leave such enduring biological traces. Now, animal models, they've been really critical for digging into the mechanisms, haven't they? The work of Michael Meany and Moshe Seif seems really important here.
Absolutely. Animal models, usually rodents, let researchers control the environment much more precisely. Dr. Meany's work with rats is a classic. He simply observed natural variations in how much mother rats licked and groomed their pups.
Just natural differences in mothering style.
Exactly. And he found this had huge lasting effects on the pup's stress response when they grew up. Pups who got lots of licking and grooming were less fearful, had a calmer stress response.
And biologically.
Biologically, they showed increased activity of that glucocorticoid receptor gene, NR3C1 in rats, specifically in the hippocampus, a key brain region for stress and memory. And crucially, this was linked to epigenetic changes.
What kind of changes?
Decreased DNA methylation and increased histone acetylation right at the control region of that gene. Basically, the nurturing care made the gene more active through epigenetic modifications.
And what was so amazing, I remember reading, was that this behavior, the licking and grooming, seemed to be passed down too.
Exactly. Female pups who received high levels of care grew up to be high licking and grooming mothers themselves. And they showed the same epigenetic patterns. Cross-fostering studies really nailed this down. They took pups born to low-licking mothers and had them raised by high-licking mothers, and vice versa. The results showed it was the early postnatal caregiving environment, not just the biological mother's genes, that drove both the behavior and the epigenetic marks in the offspring. It's a powerful example of non-genetic inheritance of behavior through epigenetics.
Wow. And Dr. Seif's work built on this?
Yeah, Moshe Seif really pioneered the field we now call social epigenetics. He used models like separating pups from their mothers for periods early in life, a form of early life stress.
And found epigenetic changes.
Yes, lasting epigenetic changes, both DNA methylation and histone modifications in important brain genes involved in stress, learning, and mood, like BDNF, AVP, and again, NR3C1. But what was also incredibly important, maybe even revolutionary at the time,
What was that?
he showed that some of these epigenetic marks and the resulting behavioral changes in the stressed animals could actually be reversed in adulthood
Reversed? How?
by using drugs that target those epigenetic mechanisms, like drugs that inhibit DNA methylation or affect histone modifications.
That's huge. It suggests these changes aren't necessarily permanent, set in stone.
Exactly. It opened the door to thinking about interventions.
Our sources also highlight Dr. Randy Jirtle's work on environmental toxicants and transgenerational inheritance. This feels incredibly relevant today with all the chemicals in our environment.
Dr. Jirtle's research has been absolutely groundbreaking here. Using rodent models, he showed that exposing pregnant females, the S0 generation, to certain environmental chemicals, particularly endocrine-disrupting compounds like the fungicide vinclozolin or DDT, could induce epigenetic alterations, primarily changes in DNA methylation patterns within the germline specifically, in the sperm of the male offspring, the F1 generation.
In the sperm, so they could be passed on.
Precisely. These altered methylation patterns, sometimes called epimutations, were then transmitted to subsequent generations F2, F3, even F4, without any further exposure to the chemical.
Wow. And these inherited epigenetic changes had consequences.
Yes, they were linked to a whole range of health problems in those descendant generations. Things like reproductive issues, increased rates of various cancers, kidney disease, immune system problems, even behavioral alterations.
So it's not just the individual directly exposed who's affected, but potentially their children, grandchildren, great-grandchildren, all through these inherited epigenetic glitches.
That's what his research strongly suggests for these specific exposures in rodents, yes.
Now, we've talked about the evidence, but let's touch on the complexities. Studying this in humans is obviously harder. And how exactly are these changes passed down?
Yeah, studying this in humans is inherently challenging. It's incredibly difficult to disentangle true epigenetic inheritance from, simple genetic inheritance, or the direct effects of the environment on the fetus in the first generation F1, or the influence of the parents' behavior and environment on their children after birth, F1 affecting F2, or just shared environmental factors hitting multiple generations.
Yes.
For a true transgenerational effect, the epigenetic information has to be transmitted through the germline, the sperm and egg cells. And as we mentioned, there's that big reprogramming event.
Yeah.
The reset.
Right. But the evidence now suggests some regions of the genome might resist this erasure, or perhaps the marks get reestablished somehow carrying a memory. Non-coding RNAs present in both sperm and eggs are also emerging as potential carriers of this intergenerational information.
Our source also brings up the weathering hypothesis. That feels like a really important concept for understanding health disparities.
It really is. The weathering hypothesis suggests that chronic exposure to social and economic disadvantage, things like poverty, discrimination, constant stress, leads to a cumulative biological burden and accelerated aging.
Or wear and tear.
Exactly, wear and tear on the body's systems. Epigenetic mechanisms are thought to be one key way this weathering occurs, embedding that chronic stress biologically. And this contributes to the health disparities we see between different groups, disparities that can persist across generations. It's not necessarily about inheriting a specific trauma mark from a single event, but more about the ongoing, accumulating impact of adverse social environments over time.
And going back to Dr. Yehuda's finding of opposite epigenetic marks in the Holocaust offspring, that really underscores the complexity, doesn't it? It's not just a simple copy-paste of the parent's mark.
Exactly. It strongly suggests a more intricate developmental response is happening in the offspring. Maybe it's an adaptation to the parent's altered physiology or behavior, or to the environment shaped by that parental experience. It could even be protective in some ways, but might also create new vulnerabilities down the line.
So it's context-dependent.
highly context-dependent. As we've seen across all these studies, human and animal, the specific genes affected, specific epigenetic marks involved, they really vary depending on the type of stressor, the timing of exposure, the sex of the individual, and probably many other factors. It's a complex interplay.
But the persistence we see in some animal models across multiple generations, despite reprogramming, that's still pretty striking.
It is. It suggests there are mechanisms for resisting erasure or for reestablishing these marks that we still don't fully grasp, maybe involving specific types of histone modifications or those ncRNAs in the germ cells. Lots more to uncover there.
Okay, so the environment, both our physical surroundings and our social world, clearly leaves a major, lasting imprint on our epigenome. Let's explore that interaction a bit more.
Absolutely. The epigenome really does act like a biological record, a mediator between our genes and the world around us. The whole field of environmental epigenetics is dedicated to understanding how these external factors modify our epigenetic marks and ultimately influence our health.
And the range of factors is huge based on our sources.
Okay.
Pollutants, diet, stress.
It really is. Think about air pollution studies are linking exposure, especially early in life, to epigenetic changes associated with neurodevelopmental issues and later risk of neurodegenerative diseases. Heavy metals like lead or cadmium, they can cause widespread epigenetic changes linked to cancers and developmental problems. Pesticides, too. Some have been shown, particularly in animal models, to cause epigenetic changes, including in the germline, affecting reproduction, metabolism, development, potentially transgenerationally, like Dr. Girdle showed. Endocrine-disrupting chemicals found in plastics, cosmetics, etc., they're known to mess with hormones, and they do so partly by altering epigenetic programming during critical developmental windows.
Even alcohol exposure before birth.
Right. Prenatal alcohol exposure leading to fetal alcohol spectrum disorders involves significant epigenetic disruption. And then there's nutrition, which is massive.
We talked about the Dutch famine, undernutrition.
Exactly. Undernutrition during development can epigenetically program that thrifty phenotype, increasing later risk for metabolic disease if food becomes plentiful. But the flip side is true, too.
Overnutrition.
Yes. Maternal obesity or a high-fat diet during pregnancy can also lead to epigenetic changes in offspring, again affecting metabolic genes and predisposing them to obesity and related issues. And it's not just calorie-specific nutrients matter hugely.
Like folate.
Folate, B vitamins, these are crucial methyl donors, the building blocks for DNA methylation. Their availability directly impacts the epigenome. Even the types of fats we eat can influence epigenetic regulation of metabolism.
And then there's the whole realm of psychosocial stress and what are called the social determinants of health. It really feels like stress gets under our skin, biologically speaking.
It absolutely does. Early life adversity, neglect, abuse, and chronic stressors later in life, like living in poverty, facing discrimination, social isolation. These experiences have been strongly linked to lasting epigenetic modifications.
In which
Often in those key stress-related genes we've mentioned, NR3C1, FKBP5, but also in genes involved in brain development, neurotransmitter systems, BDNF, SLC6A4 for serotonin transport. These epigenetic changes can contribute to a dysregulated stress response system, the HPA axis, chronic inflammation, immune dysfunction, and increased risk for both mental health conditions like depression and anxiety and physical health problems.
even aging.
Yeah, some studies link chronic stress to epigenetic markers of accelerated aging, like shorter telomeres, those protective caps on our chromosomes. The weathering hypothesis really captures this, how chronic social adversity leads to this cumulative biological wear and tear, mediated in part by epigenetics, contributing significantly to health disparities.
So putting this together, it sounds like the environment many of us live in today is radically different from the one our ancestors evolved in. And this environmental mismatch, as the source calls it, might be causing epigenetic problems.
That's a really crucial concept. Our epigenomes were shaped over long periods by ancestral environments, different diets, activity levels, social structures, stressors. The rapid shift to modern industrialized lifestyles has created a major mismatch.
Examples?
Well, the abundance of processed, energy-dense foods compared to ancestral diets are largely sedentary lifestyles versus constant physical activity. The chronic, relentless psychosocial stress of modern society versus perhaps more acute episodic stressors in the past. Our constant exposure to novel synthetic chemicals, all these represent mismatches.
And our epigenome reacts in potentially unhelpful ways.
It can lead to epigenetic responses that were maybe adaptive in the past but are maladaptive now. The thrifty phenotype is the classic example, programmed by famine conditions. It becomes detrimental when calories are abundant, leading to obesity and diabetes.
And these factors often pile up, right?
It's exactly. Especially in disadvantaged communities, people might face co-exposure to multiple stressors, pollution, poor nutrition, high psychosocial stress. These can have additive or even synergistic negative effects on the epigenome, amplifying the health burden. It's even possible, as the source suggests, that some epigenetic patterns linked to disease today are actually lingering signatures of responses that were once beneficial in ancestral environments, but are now out of sync.
This leads us to a really fascinating, maybe slightly more speculative idea from our source, the erosion of wisdom through the loss of what it calls cultural feedback loops. How might traditional cultural practices have historically acted as sort of epigenetic regulators?
Yeah, this is a really thought-provoking angle. The idea is that traditional cultural practices, often developed and refined over many generations, provided consistent environmental and social signals that helped shape and maintain adaptive epigenetic landscapes within those communities.
Yeah.
They acted as stabilizing, orienting inputs.
Like traditional diets, which we just touched on, they were so different.
Absolutely. Ancestral diets weren't just about macronutrients. They often involved incredible biodiversity, local sourcing, seasonality, specific preparation methods that might enhance nutrient availability or reduce toxins. These diets likely provided a complex mix of nutrients and bioactive compounds, things beyond basic vitamins that could directly influence epigenetic mechanisms.
For example.
Well, high fiber content promotes healthy gut bacteria, and these bacteria produce short-chain fatty acids like butyrate, which is known to inhibit histone deacetylases, enzymes that generally silence genes. So traditional diets could have directly impacted histone modifications. The loss of these diets, the shift to Western patterns, is a huge ancestral mismatch with likely epigenetic consequences contributing to chronic disease. Think of food as medicine. That concept, rooted in traditional ecological knowledge, tech, recognized the complex biochemical effects of foods, potentially including epigenetic ones.
And it goes beyond food, right? The social and emotional environment created by culture, rituals, storytelling, community support.
Definitely. Rituals, ceremonies, storytelling, these aren't just quaint customs. They often serve critical functions. Transmitting knowledge, reinforcing social bonds, providing a sense of identity and belonging, offering structured ways to process grief or celebrate transitions. They create predictable, supportive social environments.
Which buffers stress.
Exactly. They can buffer the negative physiological impacts of stress. Think neurobiologically, communal rituals or storytelling can trigger oxytocin release, promoting bonding and calming the nervous system. The predictability itself can be calming. When these practices decline or are disrupted, maybe through colonization or rapid modernization, communities can lose these crucial stress buffering mechanisms, leaving individuals more vulnerable, potentially leading to epigenetic dysregulation.
And the source also mentions the connection to nature itself and traditional ecological knowledge, tech.
Yes, tech often involves a deep, reciprocal relationship with the natural world. This isn't just abstract. In traditional societies, it meant much closer, more consistent contact with diverse natural environments.
Meaning more microbes.
Precisely. More exposure to a wide array of environmental microbes from soil, plants, animals. This is now understood to be crucial for training the immune system and establishing a diverse, healthy gut microbiome. And as we know, the gut microbiome communicates extensively with our own cells, including influencing our epigenetics.
So losing that connection matters biologically.
It likely does. Loss of tech, displacement from traditional lands, reduced time in nature. This can mean less exposure to beneficial microbes, potentially impacting immune function and maybe even epigenetic regulation via the microbiome. It also means losing access to traditional plant medicines, whose compounds might also have epigenetic effects.
So the disruption of these long-standing cultural practices isn't just, a loss of heritage. It's potentially a loss of vital epigenetic regulatory inputs, contributing to this ancestral mismatch.
That seems to be the argument. The loss of these culturally embedded orienting inputs leaves the epigenome potentially more vulnerable to the novel stressors of modern life. It could be a factor contributing to health disparities, particularly in indigenous or marginalized communities that have experienced significant cultural disruption and historical trauma. Epigenetics could be one mechanism through which that trauma becomes biologically embedded across generations.
The source even raises the idea of biological devolution, epigenetically speaking.
Right, not in the sense of genetic change, but perhaps a drift towards maladaptive patterns of gene expression. due to this chronic dissociation from the ancestral environments and practices that our biology expects, leading to reduced health, resilience. It's a provocative idea, suggesting it's not just the presence of new stressors, but the absence of these ancestral buffering and regulatory factors that drives epigenetic drift.
This paints a somewhat concerning picture, but the sources also really emphasize the inherent plasticity of the epigenome, the potential for reversal, for restoration. So can this epigenetic damage actually be reversed?
That is the really hopeful message running through all of this, because epigenetic marks are, by their nature, dynamic and modifiable. There's significant potential for change. We have those eraser enzymes we talked about.
Right.
And animal studies have convincingly shown that some stress-induced epigenetic marks and the related behavioral changes can be reversed through things like pharmacological interventions, drugs targeting those enzymes, but also through environmental enrichment, basically providing a more stimulating and positive environment.
And in humans?
Human studies are catching up. There's evidence suggesting that interventions like psychotherapy, particularly trauma-focused therapies, can be associated with changes in epigenetic markers in stress-related genes. Lifestyle changes too. Diet, exercise are showing epigenetic effects. And then there's the more futuristic stuff.
Like age reversal.
Yeah, very early research exploring partial epigenetic reprogramming using Yamanaka factors to potentially rejuvenate cells. Still highly experimental, lots of hurdles and ethical questions, but it highlights the inherent manipulability of the epigenome.
Okay, so if we're aiming for this kind of epigenetic terrain recovery, what sorts of interventions look promising based on the sources?
It seems like a multi-pronged approach is needed. One really important area is cultural and community-based restoration. For communities impacted by historical trauma and cultural disruption, revitalizing traditional practices around diet, language, ceremony, connection to land could be powerful. It might help reinstate some of those ancestral regulatory inputs and buffer stress. Strengthening community support networks is key.
And on an individual or family level?
Given the impact of early life, things like trauma-informed parenting, support for caregivers, family-centered approaches are vital. Then there are more direct biological interventions.
Like targeting the microbiome.
Exactly. Understanding that gut-brain immune epigenome axis. It opens possibilities for using probiotics, prebiotics, perhaps even fecal transplants to modulate the gut microbiome in ways that might positively influence host epigenetics.
And lifestyle, of course.
Absolutely critical. Diet is huge nutrient-dense foods, fiber for those short-chain fatty acids, polyphenols from plants, sufficient methyl donors. Exercise has well-documented epigenetic benefits, affecting muscle, brain function, metabolism, stress management techniques.
Meditation, yoga.
Yes. Practices like meditation and yoga have been linked to favorable epigenetic changes, including in stress-related genes and even influencing telomere length. And prioritizing good sleep seems crucial for maintaining epigenetic balance.
What about therapies for trauma, like PTSD?
Psychotherapies, especially those designed to process trauma, show associations with epigenetic shifts. And there's growing interest in psychedelic-assisted therapies for conditions like PTSD and depression, with preliminary research suggesting they might induce beneficial epigenetic changes related to brain plasticity and stress response.
It sounds like we need reliable ways to measure these changes and a broad view of what recovery means.
Definitely. Identifying reliable, accessible epigenetic biomarkers is a major goal, but still challenging. And terrain recovery probably isn't just about flipping individual epigenetic switches back. It's likely more about restoring a healthier, more adaptable overall epigenetic landscape. Promoting resilience.
As a holistic view.
Exactly. It requires thinking about biology, psychology, social context, cultural factors, environment, all interconnected. It's about reducing harm and restoring beneficial inputs. Maybe resilience itself has a distinct epigenetic signature we could aim to cultivate.
This has been a truly deep and really fascinating exploration. It's a lot to take in, but to recap, it seems pretty clear that our culture, our environment, even the lives of our ancestors and big societal shifts, they really can leave biological imprints through these epigenetic mechanisms.
Yeah, influencing our health, our vulnerability, maybe even across generations. Concepts like epigenetic drift and ancestral mismatch really capture that profound connection between our lived experience and our biology.
And while understanding all this might feel a bit, heavy, it's so important to circle back to that hopeful message of epigenetic plasticity. The fact that these marks aren't necessarily forever, that there's potential for change, for terrain recovery through so many different avenues. that really underscores the importance of a holistic approach to health and well-being.
Absolutely. It gives us agency individually and collectively.
So maybe here's a final thought for you, the listener, to ponder. Given that our biology carries these echoes of the past, how might we consciously shape our present environment, our cultural practices, our communities to cultivate a healthier epigenetic legacy? Not just for ourselves right now, but for the generations still to come. What responsibility comes with knowing this?
That's the big question, isn't it? Perhaps something we can explore further in future deep dives.
Definitely food for thought. Thank you for joining us on this exploration.